Organization of the neural layer of the retina. So the cells of the neural layer, photoreceptor cells that contain pigment molecules that react to light energy and convert it into electrical energy. Immediately internal to the photoreceptor layer is a layer of bipolar cells, right here. And then the innermost layer, adjacent to the posterior cavity, are the ganglion cells. And their axons extend into the optic nerve, as the incoming light must pass through the ganglion, bipolar, and then go into the rods and cones before the signal goes back out towards the optic nerve. Horizontal cells are also important. These cells are located in between the photoreceptors and the bipolar cells and regulate and integrate the stimuli from photoreceptors. Endocrine cells are found between the bipolar cells and ganglion cells and integrate visual information that is passed between the bipolar and the ganglionic neuron. And the photoreceptive cells, the rods and cones, are located right here. On the far right, or the most outer layer, we also have the retinal pigment epithelium, called the RPE. The ganglion cells and the retinal pigment epithelium RPE. The ganglion cells are filled with a pigment called melanopsin. 1 to 3% are photoreceptive, which have the following instructions. They help reset the internal clock of the cell. They help regulate the pupil size, allowing how much light comes in. And they also regulate the release of melatonin. The RPE, the retinal pigment epithelium, contains melanin and it helps trap any unused photons that are not captured by the photoreceptors. This is also where you have these granules of storage of vitamin A. Zooming into it, you can see that these are tight junctions. Within the RPE, we have small granules that can house the vitamin A, and they also help capture any additional photons that are not used by the photoreceptors of the rods and cones. Rods and cones. The outer segments of the photoreceptors extend into the pigment layer of the retina and are composed of these flattened membranous discs containing photopigments that function to absorb light, and they're constantly being replaced. The inner segments of our photoreceptors contain these organelles and connect directly to the cell body. And then there's also a synaptic terminal on the other side of the cell body that houses vesicles and it releases neurotransmitters called glutamate. So glutamate gets released over here. The rods are a little bit longer than the cones, and they have a rod shape on the outside, whereas cones have a conical shape on the outer segments. Each eye has more than about 100 million rods located in the peripheral regions of the neural layer. The function of the rods is to detect dim light, light and dark tones, or movement, especially at night. The rods converge on a few bipolar cells which synapse onto one ganglion. This allows for the control of spatial summation at both the bipolar and ganglion cells to release either more neurotransmitters or less. Cones, on the other side, occur at a density at a lot less, so it's about 100 million per eye, and are concentrated predominantly at the fovea centralis. And they provide visual sharpness and color in high intensity light. The fovea centralis is where there's a 1-to-1 ratio between each cone and to each bipolar cell and to each ganglion cell. So remember, many rods converge to many bipolar cells which converge to one ganglion, whereas in the cones, it's one cone to one bipolar cell and then one ganglion cell. Phototransduction. Photoreceptor cells synapse onto horizontal and bipolar cells. The process in rods begins with the rhodopsin compound. So going from a dark cisretinal form to a light transretinal form, depolarization occurs in the dark, meaning that the cation channels on the photoreceptor cells are open, allowing sodium to go in and calcium to go in, and that happens when you're in a dark room. When you go into a bright room, the cells are going to be hyperpolarized. The photoreceptors and channels are then closed, blocking sodium and calcium out, causing an active transduction to occur. With the light allows a hyperpolarized state of the cones, which will then cause depolarization of bipolar cells, and the action potentials are then carried off to the ganglion cells of the optic nerve. Receptive fields provide visual acuity. Receptive field is defined as all of the cells involved in the capturing of light from one single photon, including the ganglion cells and the signal that converges on. This is a contrast between the on-center and off-center ganglion cells that provide contrast and visual acuity. If everything in the middle of the view is going to be activated by the photon, then this region is going to be more focused, precise. If the peripheral or off-center is captured, then the middle is going to be negative. So as a result, it depends on which set of neurons are fired. So let's say the center cells are stimulated with photons. What's going to happen is the photoreceptive cell is going to close the cation channels, preventing any influx of sodium or calcium ions. Therefore, there's a negative sign. The negative sign indicates no ions going in. As a result of this photoreceptors not getting any ions going in, it will be hyperpolarized, which will then cause the bipolar cells are then no longer inhibited and they begin to release glutamate. So glutamate gets released over here, and glutamate will then stimulate the ganglion cells to fire more neurotransmitters to the brain for interpretation of what you see. So photoreceptors contain photopigments that capture the photons. These rods and cones contain discs that are tightly packed with the photopigment molecule opsin and the 11-cis-retinol compound. Each photopigment captures a single photon of light and converts it into energy, into a message. When light stimulates the photoreceptor cells, it stops releasing glutamate, and the bipolar cells are hyperpolarized, which will then cause them to release the neurotransmitter to send a signal to the ganglions. Scotopic versus photopic vision. Rods contain rhodopsin, scotopic vision, which functions optimally under low light conditions with shades of gray and black. Cone cells contain opsin, which are photopic vision, which function much better under bright light conditions. So the photopigment molecules in rods, which is rhodopsin, is involved in phototransduction of dim light and is most sensitive at light between the 500 nanometer range. Whereas the photopigment molecules in the cones, which is the opsin, contain either the blue, which detects the 420 nanometers, the green, which detects 531 nanometers, and the red, which detects the 558 nanometers. So you can see that the cones can pick up signals over here, the blue, green, and red. So let's take a look at how the nerve signals work at a molecular level. So we'll start with the dark pathway first, and then we'll compare it to the light pathway. So in the dark, the outer segment of the rods continuously produce cyclic GMP from GTP, guanosine triphosphate. And this reaction is catalyzed by an enzyme called guanylate cyclase. Cyclic GMP is going to bind onto the cation channels in the outer membrane of the photoreceptor, and this allows sodium and calcium to go into the cell. The cyclic GMP channels are open, causing us to have a dark current on the membrane. So there's a signal going through because ions are going in and a movement of charges along the membrane of the photoreceptor. This will then cause the calcium ions to enter in the synaptic terminal, triggering a release of glutamate over here. So glutamate is being released. It's going to bind onto the bipolar cell, causing a hyperpolarization, which inhibits the bipolar cells from releasing any more signal. So whenever the glutamate binds to the bipolar cell, there is no signal from the bipolar cell. On the right-hand side, let's say you are in the light. The rhodopsin is going to split through the bleaching reaction. The enzyme phosphodiesterase is going to break down the cyclic GMP, and therefore cyclic GMP is not going to be activated. This will then cause a lowering amount of cyclic GMP, leading to the closing of the cyclic GMP channel. Now, because this door is closed, there's no ions and no sodium going into the cell. This membrane is now not depolarized. There is no potential going through this membrane, causing this cell to be hyperpolarized, and therefore the voltage-gated calcium channels are closed and there is no glutamate being released. As a result of no glutamate being released, the bipolar cells are not going to be inhibited, and therefore they can then release so these cells are going to then release glutamate to the ganglion cells, and then when there's a sufficient amount of glutamate attached to the ganglion cells, a nerve signal is produced in the ganglion cells and is sent to the brain. So let's remember that the isomerization occurs after photon is absorbed inside the photoreceptive cells. Rhodopsin inside the rods is going to be in a bent shape in the dark. When light hits, it turns into a flat shape. So we have the cis-retinol, which is a bent shape, and then we have the all-trans-retinol, which is a flat shaped. So here are the steps of the phototransduction cascade, summarizing what we said. So let's go over the steps in the phototransduction cascade. So this is a G-protein pathway. So when light hits, the retinol cis form is going to become the trans form right here. And G-protein alpha beta gamma is going to go from a GDP to GTP. And the G-protein will then bind onto the alpha unit, causing the alpha unit to be moving along the membrane. Then it activates an enzyme called the PDE, which is phosphodiesterase. And this enzyme is going to convert cyclic GMP into 5' GMP. Because there is no cyclic GMP bound to the channel, this door is going to now be closed because the GTP is now taken away and the doors are closed down. As a result, this membrane, shown in blue, is going to be hyperpolarized. So there's no signal being released to the bipolar cells. Everything that doesn't happen here would happen in a dark room. This is what happens in the light. So now in the dark, when you turn off all the lights, the small molecule protein arrestin is going to bind onto rhodopsin, which will prevent it interacting with the G protein phosphodiesterase. A kinase is going to phosphorylate rhodopsin to inactivate it. The transducin is then turned off when the GTP is hydrolyzed into GDP and pyrophosphate, so this will then create more. And because of the transducin is unbound to the phosphodiesterase G protein, it turns it off. Because PDE is not working, cyclic GMP is made from GTP, which will bind onto the channel, causing more ions to go in. As more ions go in, the cell is considered depolarized. Then glutamate is released onto the bipolar cell, which will cause the bipolar cells to become hyperpolarized. And because the bipolar cells are hyperpolarized, there is no signal going to the brain. The visual pathways. The optic nerves converges on the secondary ganglionic axons and project from each eye through the optic foramen. They eventually congregate at the optic chiasm. This is the crossing over. So the right eye goes to the left side of the brain, and the left eye goes to the right side of the brain. These optic tracts then extend laterally from the optic chiasm as a composite of these ganglionic axons from the retinas of each eye. Most of these are going to extend to the lateral geniculate nucleus of the thalamus, where most of the visual information is processed. As that gets routed out, that then gets sent towards the visual cortex and the occipital lobes in the back for conscious interpretation of that visual stimuli. So reminding yourself that the right and left eyes do have overlapping visual fields, and the brain must be able to unite them to create one image.